Cookie Smokes. In this video, I'm going to be talking to you about prokaryotic cells. So that word prokaryote comes from the Greek, meaning before, before, and nut or kernel, which is what they are using to describe the term nucleus. So prokaryotes evolve before nuclei. Prokaryotes are the oldest, they are the most ancient form of life on Earth, and they are also the most abundant form of life on Earth. So prokaryotic organisms appeared on Earth about 3.5 billion years ago. This is a very long time, and they lived all by themselves for about 2 billion years. So it was a long time before eukaryotic cells came about. The first sort of prokaryotes were probably cyanobacteria, so bacteria that are able to perform photosynthesis. So these prokaryotes, they're floating around, they're very simple. What are they like? First of all, we now split all life into 3 domains: the archaea, the eubacteria, and the eukaryotes. So both the archaea and the eubacteria are prokaryotic forms of life. Let's look at the archaea first. So archaea are an incredible group of organisms. So these are prokaryotic, and they tend to be extremophiles. These are organisms that are capable of thriving in really extreme conditions that we would not be able to survive in. So for instance, some archaea can live in extremely salty conditions. These are known as halophiles. So you can find them in places like the Dead Sea or the Great Salt Lakes, where no other organisms could survive, because they have evolved adaptations to allow them to cope with these high salt concentrations. In other organisms, these high salt concentrations would draw water out of the cells by osmosis and cause those cells to die. But the adaptations that these halophiles have keep them alive. Some archaea can survive in extremely hot environments. These are known as thermophiles. Some archaea can survive in really high pressures. These are known as barophiles. Some archaea are known as methanogens. These are anaerobic archaea that actually produce methane as a byproduct of their metabolism, as opposed to using oxygen. They use carbon dioxide and hydrogen in order to release energy. These are really interesting. They are obligate anaerobes. That means they can't survive in anaerobic conditions, and they get poisoned by oxygen gas. So one place that you're probably really familiar with methanogens is that they are found in the gut of some animals, such as sheep and cattle. So you probably already know that one of the problems with farming cows is they produce huge amounts of methane that they release into the atmosphere, and that methane is produced by the methanogens that live in their gut. But we often forget that those methanogens are performing a really important role within the gut of cattle, because they're involved with helping with the digestion of food. Without the methanogens, the cattle wouldn't be able to digest their food, they wouldn't be able to grow. Each year, methanogens release around 2 billion tons of methane into the atmosphere, which, as you know, is pretty problematic because it acts as a greenhouse gas. One more fact about archaea before we move on, and that is that they are more closely related to eukaryotes than they are to bacteria, which is really fascinating, because I think people tend to put the archaea and the eubacteria in one group and then the eukaryotes in another group over here. But it doesn't work like that. The way that evolution has occurred, archaea are more closely related to us than they are to bacteria, which is pretty fascinating. If this level of detail with evolution is something that really interests you, an author I would really recommend reading is a guy called Nick Lane. He's written a bunch of books that are really fascinating, but the one I think that would be most appropriate in this topic is one called The Vital Question: Why is Life the Way it is? I would highly recommend that one. So prokaryotes are the most abundant organisms on Earth, and they come in some pretty different structures. So we can see cocky bacteria. These are spherical. Bacilli bacteria, these are rod-shaped. Spirillum bacteria, these are spiral. And Vibrio bacteria, these are comma-shaped. So for instance, a very well-known rod-shaped bacteria is Yersinia pestis. This is the bacterium that caused the plague. In terms of cocky bacteria, you will have heard of streptococcus, which causes a sore throat, strep throat. What other cocky bacteria might you have heard of? For instance, there's Diplococcus pneumonia that causes pneumonia, and there's Neisseria gonorrhoeae, which causes gonorrhea. There's also Staphylococcus, which you've probably heard of when you learnt about MRSA. Staphylococcus can cause wound infections, it can cause toxic shock syndrome, and it can cause food poisoning. Prokaryotic cells are significantly smaller than eukaryotic cells, and they have a diameter of between 0.1 and 5 micrometers, compared to eukaryotic cells that have a diameter between 10 and 100 micrometers. So, they're much smaller. They have no membrane-bound nucleus. Their genetic material is free-floating in the cytoplasm, and they have no internally membrane-bound organelles. So, as opposed to eukaryotic cells, they don't have mitochondria, they don't have rough endoplasmic reticulum, they don't have chloroplasts, they don't have lysosomes, they don't have any internally membrane-bound organelles. So they're super, super tiny organisms that are very, very simple on the inside. So they have this free-floating DNA, which is circular as opposed to linear. They have a cell wall, and many eukaryotes have cell walls, but the cell wall in prokaryotes is made of murine, which is a glycoprotein. They have a cell surface membrane, which is very, very similar to a eukaryotic cell surface membrane, and they have cytoplasm, just like the cytoplasm inside a eukaryote. They have ribosomes because they need to do protein synthesis, but the ribosomes that they have are much smaller than the ribosomes in eukaryotic cells. So they have smaller ribosomes. Some prokaryotes have flagella, but those flagella are smaller than the flagella that you would find in a eukaryote. So we know that a flagella is a long structure that rotates to enable a cell to move, and you probably know about flagella on sperm cells, and many prokaryotes use flagella in order to move. So the DNA in a prokaryote, it's circular, and there tends to be one loop, but secondary strands can form. These smaller loops of DNA that are not part of the main circular DNA are called plasmids. So sometimes prokaryotic cells have plasmids, sometimes they don't. Many prokaryotes also have a capsule, which is made up of it's kind of called a slime layer. And essentially, it's there to protect them from a number of things in their environment, but it can be there to protect them from an attack by cells of the immune system, for instance. Not all prokaryotes have a slime layer, and even those that do don't necessarily have a slime layer throughout their whole life cycle. The other thing that we see in a prokaryotic cell is something called a mesosome. A mesosome is an extension of the cell membrane. This is actually a really important feature of prokaryotic cells because they don't all have these organelles on the inside that increase their surface area for metabolic reactions to occur. They have to increase their surface area in another way, and that's how they do it, by having these long infoldings of their cell surface membrane, and the enzymes involved in the metabolic processes that go on inside prokaryotic cells are often attached to the mesosome. So let's put all of that information together and have a look at the structure of a generalized bacterial cell. And let's just be aware this is like a generalized bacterial cell. Lots of bacterial cells have their own specific features. Bacteria replicate by binary fission. Binary fission is a simpler form of mitosis. Essentially, this means that they replicate their contents, including their DNA, and then they split into two cells. Under ideal conditions, bacteria can do this extremely quickly, but the rate at which they do this is dependent on the organism, the temperature of the environment, and other conditions. E. coli, under perfect conditions, can do this every 20 minutes. So, because bacteria don't undergo sexual reproduction, they only undergo asexual reproduction, they don't have all the benefits of mixing DNA from two organisms in order to create variation in the next generation. All of their variation is created due to mutations that occur in the DNA replication. But there is another way in which they can share their genetic information. This process is called horizontal gene transfer. Essentially, what happens is these plasmids inside the bacteria, these can be replicated separately to the main chromosome. If two bacteria come across each other, they can grow a structure, which is known as a pilus, that joins the two bacteria temporarily, and they can swap their replicated plasmids. When this occurs, the bacteria have gained genes. As I mentioned earlier, these plasmids, they often contain genes for things like antibiotic resistance, and this is one reason that antibiotic resistance can spread so quickly amongst bacterial populations. So, in this video, we've looked at the generalized structure of prokaryotes. We've looked at their DNA and what's special about their DNA in comparison to eukaryotes. We've also looked at how prokaryotes are classified as living things. And at this time, that's pretty much everything that you need to know about prokaryotes, so well done.